Dr. Marshall. He's going to talk to us about the development of drugs that strengthen synaptic connections for the potential treatment of Angelman syndrome, the role of BDNF, brain-derived neurotrophic factor.
Thank you. I don't know if you saw that photograph of me. I had more hair back then, so it's a pretty old photograph. But today I want to talk about some other work that we're doing at Brown in collaboration with Dr. Huang's lab and Xin Yang on the development of drugs that enhance the strengthening of synapses. And these drugs act to increase the activity of brain-derived neurotrophic factor. So what is oh, no disclosures. So what is brain-derived neurotrophic factor? It's a growth factor that's really important for the strengthening of, for synaptic plasticity. And synaptic plasticity is the ability of neurons to change their strength. And BDNF plays a role at the synapse. The synapse is the contacts where nerve cells interact. Here we have a presynaptic cell. It sends electrical impulse down to the terminal, where at the terminal, at excitatory neurons, you release glutamate, the amino acid. That glutamate then activates the second cell, as shown here, to generate another impulse, and so on and so forth. So you can set up circuits, networks of activity that are important in learning and memory, in speech, in walking, in remembering what you had for breakfast, that type of thing as well. So unfortunately, in Angelman syndrome, these synapses appear to be weaker. They don't strengthen in the correct way. And we believe that's due to deficits in the ability of BDNF to signal. And this is shown here on this slide. So this is just a cartoon, again, presynaptic cell. You have these vesicles containing the transmitter glutamate. When an impulse comes down, glutamate is released. It diffuses where it interacts with the glutamate receptors. And in the normal situation, the growth factor BDNF will bind to its receptor and activate various signaling pathways. And it activates these enzymes, one of which is the PI3 kinase enzyme, which then activates an enzyme called mTOR. mTOR has a really important function in that mTOR, is important in the expression of proteins, receptors that are important in strengthening this particular synapse. mTOR also has another role that I'm going to talk about today in terms of inhibiting the breakdown of these important proteins in the strengthened synapse, a process called autophagy. So we found that this signaling pathway is regulated by a protein called PSD95. It's a very abundant protein found at synapses. Unfortunately, in Angelman syndrome, when UB3A is absent, you increase the levels of a protein called ARC. ARC is normally a very transient protein. It's expressed and removed. In neurons in Angelman syndrome, it's more stable out in the dendrites. And it interferes with the ability of PS95 to signal. It sticks to PS95 and disrupts the ability of BDNF to activate this signaling pathway. So we set out can we develop a drug that would disrupt the interaction of ARC with PS95? And so we developed a compound. This is our most recent, our most recent compound called SYN3. SYN3 binds to PS95, disrupts ARC. So now that when BDNF binds the receptor, now PS95 can associate, and then that will restore this signaling, such as CAMK2, which is really important in stabilization of glutamate receptors at the synapse. Okay. SYN3 has very high affinity for this protein called PS95. We designed it so it binds to 2 different sites, so that gives it very high affinity, high specificity for this particular protein. okay? And we have some of the affinities shown here. It's actually a peptomimetic. It has a cyclic type sequence to increase stability. And this is the SYN3. It has a pretty low affinity. The lower the number, the greater the affinity. We've also designed another compound called DSYN3, which is more stable in plasma. So when you inject the drug into a mouse, for example, or hopefully eventually patients, this drug will be more stable. Does this drug cross the blood-brain barrier? We've heard about this, and we've designed it adding a cell-penetrating peptide. It has a polyarginine cysteine type peptide. We've attached it to this compound, and this works really well at getting this compound across the blood-brain barrier, and that's shown here. This shows the concentration of this drug in the brain through a microdialysis type approach. So within about 30 minutes, that's when we first measure it, we can see it in the brain at fairly high concentrations, and then gradually the DSYN3 decreases over time, over many hours, so 5 to 8 hours. If we actually give a higher dose, this is actually a really low dose, if we give a higher dose, it would probably stay in the brain for much longer. The SYN3 also works really well, gets into the brain, not quite the same concentration as DSYN3, and also maintains for a long time. We actually do find that the DSYN3 is more beneficial in treatment of mice. So in this, we've injected the drug. We've also done tests where we can give the drug subcutaneously. So for example, diabetics with type 1 diabetes, they inject insulin. It's very simple. Kids do it. It's a very easy way to deliver a drug. We've also tested intranasal delivery. I haven't shown it here, but the intranasal actually looks even better. It goes way up and then comes down again. So intranasal is another easy way to deliver these drugs to patients. We're also very interested in developing an oral approach. So you may know about Ozempic. Is a cyclic type there's a peptide-like sequence for diabetes. It's kind of similar to our compounds, but you can take it orally. So you can make these peptides orally available, and that's something we're working on. So do they work? Do they actually affect BDNF signaling? This is an experiment that I did in collaboration with Dr. Huang's lab, and Xin Yang in his lab did this experiment. He took human neurons, and when you stimulate human neurons with this growth factor BDNF, you get a very nice signal. This dark band indicates that this protein is active. This protein AKT is in this signaling pathway. So the dark band indicates that it's very active. BDNF will activate this protein. When you knock down UB3A, notice that when you add BDNF, you don't see any activity of this protein. And any of these other, we've also looked at these downstream signaling proteins and also mTOR. mTOR is also deficient. When you add back DSYN3, you see that you restore normal signaling. Okay? So the drugs work really well in restoring in signaling in these in vitro systems and in vivo, as I'll show later. One thing I wanted to talk about hasn't been talked a lot about in terms of Angelman syndrome. I mentioned that mTOR is important in making new proteins, new receptors to strengthen synapses, but mTOR also inhibits the breakdown of important proteins like these scaffold proteins like PS95. This process called autophagy is also important in the breakdown of synaptic vesicles. So that really weakens the synapses. It makes it very hard to get the strengthening of synapses. And we wanted, since mTOR is defective, we wanted to see, do we see increased autophagy? So autophagy is, as I mentioned, this process where various proteins get targeted for degradation through an organelle called the autophagosome, as shown here. They're taken to the lysosome, which is basically the waste disposal system of the neurons. Unfortunately, these proteins get taken there, they get broken down, and we find when we examine autophagy, we actually see enhanced autophagy as we would expect. Because normally mTOR will inhibit this autophagy. And in Angelman syndrome, you get increased autophagy. Actually, UB3 acts as a brake on autophagy. And since UB3 is not there, you increase autophagy. But these compounds, luckily, decrease autophagy and help to maintain these proteins. And that's also seen in the mouse. So if you look in the mouse, look at the expression of these proteins. In the Angelman mouse, you see decreased levels of synaptic proteins, which are important in formation of synaptic vesicles and the release of the transmitter. And also postsynaptically, you see decreased levels of PS95, other proteins like SHANK3, and they're also decreased. But when you add back our drugs, you see increased levels of these proteins. Okay? So we think that we've restored everything that's necessary to get learning, to get long-term potentiation. And as shown in the next slide, we do get LTP. LTP is a molecular basis of learning. And basically, as I mentioned, if you stimulate one neuron and then record another neuron, if you stimulate with the correct activity, you will strengthen that neuron, which is what we want to do. And that happens in normal mice and people. So if we look in, say, a wild-type mouse, when you look at activity in the postsynaptic cell, you stimulate with activity, you get a stronger response, as shown here. In Angelman mice, that doesn't happen. You don't strengthen those synapses. You stimulate, and you notice that the response stays right at baseline. When we give our drugs, we inject the drugs into the mice. This is the AS mouse model. We inject SYN3 and DSYN3. They both give really nice LTP, which tells us that they're actually strengthening the synapses. The other interesting thing is that you can inject the drugs and wait 3 or 4 days and then examine if you get LTP, and we still get LTP. So that shows that these drugs, even though they have a fairly short half-life, so they're in the system, they get into the brain pretty quickly, they reset the brain, they're also got rid of in the rest of the body, so they won't affect the rest of the body. But they will have these long-lasting effects. So we can imagine giving these drugs potentially to patients maybe once a week, maybe twice a week, depending on the dose, giving them sub-Q or intranasally or even orally. Okay? And then finally, do they have any beneficial effects? So we've done all these experiments in the Angelman mouse model. Well, I haven't done it. This was a collaboration with Jill Silverman's group, who is an expert at doing these types of behavior, particularly, and Emily Hui, who's a PhD student in her lab, did all these studies, and they got some spectacular results. One of the most surprising results was these compounds decrease seizure susceptibility. So of course, Angelman kids have many have intractable seizures. We need better drugs to treat those seizures. And we were really surprised that these compounds are really effective. So in this experiment, for example, looking at tonic-clonic seizures, we give a drug that induces seizures, and the Angelman mice are very susceptible. You get really awful seizures compared to, say, wild-type mice, the time it takes to induce seizures. When you give our compounds, both SYN3 and DSYN3, you get seizure activity similar to wild-type. So we can imagine that these drugs could be really useful for adults in terms of reducing seizure activity. In terms of learning, Emily did this. In this case, she did a novel object test. This is where the mice on day 1 get to play with 2 objects. And then on day 2, you remove one of the objects and add a new object, which is a novel object. And mice are very inquisitive. They will go and explore the novel object. Unfortunately, Angelman mice don't do it. They don't remember that this is a novel object. And incredibly, when you inject our compounds, now the Angelman mice can remember the novel object, showing that these compounds do improve learning and memory. Okay? And then Emily also looked at motor learning. So in this case, we use a rotorod. So we put the mice on this rotating rod, and the wild-type mice just stay on and on forever. The Angelman mice haven't got the same coordination, so they fall off the rod. So we interestingly found that the SYN3, this is wild-type on the rotorod, SYN3 didn't really work so well. The DSYN3 did have a very significant effect. It didn't improve performance as well as the wild type, but it did have this sort of intermediate effect. And I should point out that these are mice, they're like 2 to 3 months old, so that would be equivalent, I'm guessing, to a human who's about 20 years old. So potentially, these drugs could be really useful in adults in improving their motor performance and coordination, which I think would be really beneficial. This is just summarizing the results. It just shows the results in the novel object. We've done similar studies with an older drug, our CN-2097, that was just published in Progress in Neurobiology. It talks about all the autophagy work that I just mentioned for the scientists who want to really look at it and criticize it. The drugs increase motor performance. I would imagine if you treat kids when they're much younger, say 1 or 2 years old, these drugs could actually restore complete motor behavior. We've seen that in this paper when we tested younger animals. Okay? And then most interesting is the drugs reduce seizure activity. So this could be a really useful drug for treating epilepsy as well, which is pretty awful. Finally, I just want to acknowledge the people who did the work, particularly Jill Silverman, who's been fantastic, and Emily, who's been incredible, generated these incredible results. It's nice to have an independent lab. You know, we've done these behavioral results in our lab, but it's nice to have an independent lab validate our results because they're pretty amazing. In my lab, Manju Bhadotti did all the LTP studies. We do a lot of structural work, which I didn't talk about, with Mandar Naik. I probably mispronounced his name. I'm trying to understand exactly how the drugs interact with the drug target. I have to thank Alvin Huang, who gave the talk before me, and particularly Dr. Xin Yang, who spoke yesterday, who's helped tremendously on the making these eye neurons, these embryonic stem cells, converting them into neurons. Xin also helped us a lot with the autophagy work as well. I also work with a lab in my old postdoc in China, who also, I've encouraged him to do some work on Angelman. And all of the work on the SYN drugs that I just talked about was funded through FAST. None of this work would have been accomplished without FAST funding, so I really appreciate that from all the parents and the fundraising. That's it. Thank you.